for today is Guru Prasad. The topic for today's session is the role of antibiotics and epigenetic alterations in genesis and prognosis of carcinoma.
In microbiology. Then I did a PhD in ocular tumors. After that, I've been working on the epigenetics. After that, I've shifted my research to microbiome and cancer. So before going into so I have two divisions of my presentations. One is the role of epigenetics discussion. After that, I'll go for the microbiomes and cancer. So in my PhD and post-PhD, I did my work on ocular tumors of surface squamous cell carcinoma. I have seen the role of DNMT3L gene and its functions, as well as I'm going to talk about the promoter methylation status of the DNMT3L gene in squamous cell carcinoma of both ocular as well as esophagus. So what is the OSSN? We call it as a normal term. It is called OSSN. It's ocular surface squamous cell carcinoma. So it is a lesion appearing on the eye, but it extends up to carcinoma up to from dysplastic lesions. It has been spectrum of dysplastic as well as carcinomatous lesions of the ocular surface. This is the incidence and associated with the lower age, xeroderma pigmentation, all that stuff. Then coming to the epigenetics, so there, as we all know, till now we discussed altered methylation status leading to the cancer formation, which leads to hypermethylation of the tumor suppressor genes as well as the protoncogenes. The studies have been carried out to find out the methylation genes like DNA methyltransferase 3A and 3B and histone acetylase. So one study which has been we found previously, which has been published in CDFD, is overexpression of DNMT3 leads to increase in the cell proliferation. So coming to what is DNMT3 gene, it is a present on chromosome. It is devoid of any catalytic activity. It interacts with the 3A and 3B and promotes the de novo methyltransferase activity. So there are strong correlations between the loss of methylation of DNMT3L gene in the promoter region and the cancer genesis. So loss of methylation has been noted in the cervical carcinoma. As the similar aspects of squamous cell carcinoma have been noticed in cervical cancer, we applied this loss of methylation or hypomethylation of DNMT3L in our ocular tumors. So these are the different studies which are which are saying that the role of DNMT3L has been associated with the carcinoma. So similar epigenetics changes, what we have seen in our cervical tumor, we applied to the ocular surface squamous cell carcinoma. So it's just pilot study. So we have taken 6 ocular tumors and 7 normal conjunctiva. Then this is all the techniques we have followed: DNA extraction, bisulfite conversion, and the cloning and all expression we have studied. So this is the biometric data, and so this is all the mechanisms what we have done. If you see here the methylation status, I think I can explain better in this way. So it's a degree of methylation. So first 7 samples, if you see, here is the conjunctival CPG islands. 11 CPG islands were studied in the promoter region of DNMT3L, and the similar CPG islands what you see in the squamous cell carcinoma. So green color indicates the 35% of methylation, yellow indicates 60, and red color indicates 100% methylation. So compared to that of the conjunctiva, there is a loss of methylation we see in the ocular tumors. So we find the significant loss of methylation in the eye in CPG island of CPG in 6 as well as CPG 9. So we can predict. So I'll come to the discussion later. So based upon the similar aspect, what we got the results in ocular tumors, we applied on the esophageal squamous cell carcinoma. So this is the controls, as well as this is the, okay, sorry. Coming to degree of methylation in esophageal carcinoma, hypomethylation has been noticed in the 4th, 5th, 12th, and 6th are statistically significant compared to that of esophagus normal tissue. So we found a lot of statistically significant loss of methylations in squamous cell carcinoma of the esophagus compared to that of. So coming to the various degree of loss of methylation has been noted in DNMT3L region of both OSSN, but the 6th CPG could possibly act as a suitable marker using a methylation-specific PCR. So similarly, in squamous cell carcinoma of the esophagus, we have noticed a lot of significant methylation loss in all these CPG things, but fifth CPG could possibly act as a suitable marker for losing, using methylation-specific marker. So coming to the discussions, the increased level of DNMT3L has been noticed, and subsequent degradation of subset of genes is due to increased stimulation of DNMT3L. So this is a part of epigenetics what we have studied. So as a microbiologist, I was much interested in microbiome and cancer. So it plays a lot of important role associated with various cancers, role in diagnosis, development, progression, and treatment of cancer. So similarly, as cervical cancer is related with the HPV, we also studied the role of HPV in ocular surface squamous cell carcinoma. So there are controversial results. The few studies have shown that HPV has been correlated with the OSSN, and similarly, that much studies have negated the role of HPV in OSSN. So in India, that time that study has been not done. So we have taken up the study and we have used the two PCRs, in situ hybridization, as well as the polymerase chain reaction we done on the biopsies. We evaluated the presence of HPV. So around we took around 50 cases and 50 samples, and we couldn't find any. We have not find any presence of HPV in the specimens. This is the positive controls what we are seeing here. Similarly, in situ hybridization also, we can able to demonstrate the presence of DNA in the cells, in situ cells of cervical tumors, but we could not able to identify any HPV. So we say that the studies showed that absence of HPV 16 and 18 in OSSN cases. So presently, I'm working on the predictive marker of the colorectal carcinoma. As you know that colorectal cancer is the fourth leading cause of death. Early detection is the most urgent need in the public health. Diagnosis is mainly based upon the colonoscopy, and there is urgent need of diagnostic predictive marker. Here, when studying up the literature, I came to a notice that E. coli B2 stain, which is consists of a PKS island, which produces a genotoxin called colibactin, which has an ability to destroy the DNA break, and promotes the tumorigenesis. Till now, E. coli, we used to think it is the normal flora of the intestine and it has caused the diarrhea. But few studies have shown that E. coli has an ability, few strains of B2 we have ability to produce genotoxin and able to break the DNA and promote the tumorigenesis. So cells infected, so several studies have been have shown that cells E. coli producing the senescence as well as PKS positive E. coli can act as a promoter for the CRC genesis. So studies have found colonic lavage have been positive for PKS, E. coli, fecal specimen have found to be positive for the PKS gene cluster. So my aim was to establish the PKS positive E. coli as a putative non-invasive marker for early identification of CRC. To evaluate the PKS positive E. coli in tumor tissues and to establish PKS positive. So these are all my aims to early prediction of the colorectal carcinoma. So I just started my work. So we screened the 40 E. coli. So before screening the E. coli in fecal matter, I just want to evaluate the PKS positive E. coli arise from the E. coli isolated from several infections. What we thought, we found around 10% of cases which are unrelated to the cancer, also showing the presence of PKS island as well as the colibactin gene. So one more research I'm planning to do, this is future planning of what I'm doing. Symbiotic bacteria tumor initiates its progression and interference in the treatment. I'm planning to study the presence of intratumor bacteria and its correlation with the cancer. Tumor with vascular hyperplasia, aerobic glycosis promote the intratumor bacterial multiplication. It affects the tumor microenvironment, promotes the metastasis. So these are two of my projects which I'm planning to do. So intratumor bacteria promotes the mutation rate, regulates the oncogenic pathways, and intimate the inflammation. So rich and diverse bacteria is located in the tumor tissue. So I'm planning to work on a fusobacterium, which is a resident of cancer, and has an ability to promote the genotoxin, and is one of the main factors which initiating the metastasis. So CRC is known to second leading death, as I told you. So on the CRC, I'm planning to study the presence of colibactin gene E. coli as well as the presence of Fusobacterium intratumor bacteria, which have an ability to promote the metastasis in CRC. So this is my study, detection of genotoxin gene in Fusobacterium nucleatum. It acts as a prognostic marker. Thank you.